So it's too fun, no? There's all these colors of chalk, so I'm going to try them out. Okay, so now before we go ahead, so I kind of gave you a trick problem, and I don't know whether you fell for the trick or not, but I said something like, I said that these researchers went to this island and they found 10 white bears and 13 black bears, right? And then I said, can you test Hardy-Weinberg equilibrium? Actually, with this information, you can't, because you don't have enough information to actually estimate allele frequencies. You can do the square root of 10, but that's not the right way to do it. The right way to do it would be, you have to have the genotypes, right? If you had AA, AB, BB, so for example, since they know it's a MC1R mutation, right? They could use PCR to amplify the MC1R locus, sequence it for all these individuals, and find out how many are AA, have 2 copies of the mutation. Say there are something like this, okay? Once you have this, then you can estimate this is the observed, right? Why is this observed? This is the observed genotype frequencies. This is the observed phenotypes. But this is actually a composite of these two, right? So you don't know what the observed genotype frequencies are unless you actually genotype the individuals and get them. That's still the observed genotype frequencies. To get the expected frequencies, what would you do? What you had suggested, 10 plus 1.5 by 23 is frequency of A, right? And then you could get frequency of B is 1 minus A, that is valid anyway, and then you would get A square, 2AB and B square, right? And then you calculate the chi-square value, which is observed minus expected whole square by expected, is that right? I think so. And you look at the chi-square. How many degrees of freedom do you have here? What chi-square table would you look at? You have 2 degrees of freedom because you can be 3 genotypes, but if you know if it's this or that, if it's not this or that, it's the third, right? So it's basically the number of genotypes minus 1, and so you can then look at whether this chi-square value, right, which you calculate, this is observed, this is expected, you calculate chi-square value, you look it up in 2 degrees of freedom to see whether this population deviates from Hardy-Weinberg, okay? That's how you do it. So you cannot do it unless you have the genotype frequencies. Without genotype frequencies, just with phenotype frequencies, unless you know it's an additive allele where there are 3 phenotypes, you can then do it, but if you don't know that, you can't. Okay. So now, someone talked about inbreeding. And inbreeding is an interesting case. We'll find out why. So colloquially, we define inbreeding as mating between relatives, which it is. That's not an incorrect definition. Someone talked about inbreeding depression. I want to point out, very important, that inbreeding depression is different from inbreeding. Inbreeding depression is the loss of fitness because of inbreeding, okay? So it's a measure of fitness in some sense, okay? So now, inbreeding per se is mating between relatives, yes. But in a population genetics context, inbreeding is actually identity by descent. So remember, your pool of gametes you're going to be pulling out gametes to assign them to individuals. Because this is an infinite pool of gametes and you're sampling with replacement, that means in some sense it's such an infinite pool that you're theoretically putting whatever you pulled out also back. You're not changing the allele frequency based on what you're pulling out. There's a chance that you will sample the exact same allele twice. Not just an A, not just a B, but that exact same A. Okay? So that is identity by descent. The same exact allele, it sounds a little odd, but because it's theoretical, the same exact allele, the same copy of that allele, individual has 2 copies of the same exact allele. Okay? So that is called identity by descent. You could get 2 A's which are from different parents. That is called identity by state. Okay? And that's not inbreeding. There could be 2 individuals who both have A's. You happen to pull their A's and you get 2 A's, right? This identity by descent, the frequency or the probability of identity by descent is called F. That's inbreeding. Okay? So now, imagine a population. How does inbreeding then change allele frequencies? Let's think about this. So you have a population. Okay, where you have individuals. So the first allele you're going to pull out, there's no chance of it being inbred, no? I mean, it's the first one, right? Only when you're going to pull out the second one, it could be inbred or outbred, not inbred. So suppose this is your first allele, and now you're going to pull out a second allele. So now in your first allele, there's A1 and A2 of frequency P and Q. Now though, for the second allele, which is on this side, you have 2 possibilities. Outbred and inbred. If the chance of inbreeding is F, the proportion of this will be F. And what about this? Should be 1 minus F. If you're not inbred, the probability of that is 1 minus F. Now within this, there is A1 and A2, right? So you can have, and this is P, this is Q, so you'll have P square 1 minus F, that is the proportion of homozygotes that are not inbred, identity by state, but not descent. They're homozygous, so you would immediately think they're inbred. Heterozygous is definitely not inbred, right? 2 different alleles, they cannot be the same. They cannot be because of the same parent, right? So they cannot be the identical same allele, right? Here you have PQ 1 minus F. Similarly, PQ 1 minus F. Q square 1 minus F. And here you have PF, here you have QF. Okay? That proportion of the alleles is inbred, right? So now, if you wanted to look at the frequency of P, it's P square 1 minus F plus PQ 1 minus F plus PQ 1 minus F, right, by 2, because you have to take half, right, plus PF. Those are all the P's, right? P square 1 minus F plus 2PQ 1 minus F plus PF I made a mistake. Oh, sorry. So to get the proportion of homozygotes, that's big P, not small p, that's p square 1 minus f plus pf, right? Heterozygotes is 2pq 1 minus f, and these other homozygotes is q square 1 minus f plus qf. So now, imagine if there was no inbreeding. What was this? No inbreeding, this was just p square, 2pq, q square, correct? So what has happened here? What has happened because of inbreeding? What has happened to the heterozygosity? What has happened to the homozygosity? Heterozygosity has decreased, right? Because it is 2pq minus 2pqf, right? So 2pq minus something, whatever f is, if f is more than 1, less than 1, 0 to 1 is what f will be, it's going to decrease. In fact, if f is 1, which is selfing, what will heterozygosity be? It will be 0, right? So, what does inbreeding do? In the offspring, these are the offspring, no? These are the offspring. It decreases the proportion of heterozygotes, correct? Okay, but if we want to calculate P, again, P square 1 minus f plus pf plus fpq, na? fpq sorry, pq into 1 minus f. So if you take p square, if you take 1 minus f common, sorry, I should use a white chalk. Okay, so if you take 1 minus f, p square plus pq, I think you guys can do this on your own time, but when you simplify this, it's basically equal to p. Okay, we'll have to substitute 1 as p minus q and all that stuff. And if you simplify it, it basically equals p. So the frequency in the gametes doesn't change. But in the adults, because of inbreeding, there's a apparent loss of heterozygosity. Okay? And this is lost by this term 2pq into f. Okay, so now
when it's inbreed, will it be pf or p square?
Which one? P into p, yeah. No, p, so this is the first allele. Okay, so that has a frequency p. And I'm going to pull out one more p, which is not inbred. So what is that? One more p is p, and it is not inbred, which is 1 minus f. So this is the first allele, this is the second allele. So to get them both, I multiply. So p square into 1 minus f. P into 1 minus f.
I think it will be p square f in the third entry.
No, because what is left is f, the proportion which are not inbred. It's not, already you have p. The proportion which are not inbred is f. Okay? So I'm going to just quickly talk about migration, and then I will go to drift and talk about, so here I'll talk about FST, and then I'll talk about drift. And in both cases, so basically migration can be looked at theoretically, which is basically if you have, say, an island, it's an island, and there's a continent, fine. The allele frequency here is Qm, the allele frequency here is Q0, okay? So, migration rate is M, this is 1 minus M, individuals on the island are natives. On the island, total number of individuals is 1, proportional. 1 minus M are natives, M are migrants. Okay? So, after one generation of migration, what will be the allele frequency here? Initial allele frequency on the island, Q0. Initial allele frequency on the mainland, Qm. How will you calculate that? All the Q alleles on the island are coming either from the natives. So what is allele frequency of the natives? Q0 into M? Oh, sorry, Q0 into 1 minus M, correct? Agree? Q0 is the allele frequency of 1 minus M individuals, right? Plus what? Who are the rest? M individuals. What is their allele frequency? Qm. M into Qm. Okay? And what is this? Q1. After one generation, frequency of Q on the island. Right? Yeah.
What is this 1 minus M with that arrow represent?
1 minus M with the arrow. Oh, it's just that this many individuals, 1 minus M individuals are here only. So there are total 1 individuals, proportionately, of them M are migrants, 1 minus M are here only. They're staying here, they're not going anywhere, they're not going back. That could make it a bit more complex if N were going back. Yeah? This is the most simple possible model of migration, unidirectional gene flow, right? Mixing. So then, what is delta Q? So we started out with the question, how does migration change the allele frequency? What is delta Q? Sorry? Q1 minus Q0, which is, go ahead, you had a question?
Sorry, but can you repeat the inbreeding concept? I don't understand how you mean to say it.
Okay. So, you're going to pull 2 alleles. So the first allele, this is your pull. You pulled out the first allele, the independent events, right? So the first allele, the probability of pulling an A1 is P, the probability of pulling an A2 is Q. Okay? Now this A1, you're going to pull another allele. Now this, the other allele could be either outbred, right? And that is 1 minus F, proportion of alleles are outbred, right? So basically, of these outbred alleles, you could pull an A1 or an A2. Okay? So, the probability of pulling a A1 that is outbred is P into 1 minus F. Okay? So you've pulled first a P and then another P. So P square into 1 minus F. P into 1 minus F. Similarly here, you pulled a P, you pulled a Q which is outbred. So P into Q into 1 minus F. Okay, this is the same as this. You pulled a Q, you pulled a, you pulled a, the second one is, this was first allele is A2 and then you pulled a P. Here, first allele is A1 and then you pulled a A2. Here you pulled two A2s. Okay, so now that is the outbred individuals. For the inbred individuals, you don't do P square because the probability of being inbred is F. Of pulling the same individuals twice is F. And that's because of some mating thing. Okay? And the chance you will do it for the P for A1 is PF. The chance you will do it for Q is QF. Got it? Okay, so back to this very simple model. There's a continent, there's an island. The continent has an allele frequency of QM, or mainland, sorry. And the island has a frequency of Q0. And individuals are moving at a rate of M. So after the first generation of movement, so by the way, at t equal to 0, what is the frequency of Q on the island? Huh? Frequency of Q on the island? Q0, right? Okay, people are asleep. I'm losing you. What's happening? Q0 is the frequency before migration, correct? Now, what's happening? Migrants are coming, fine. So now you have a mixture. In your, after one generation, you have the natives and you have the migrants. The natives, how would you calculate the allele frequency? It's just arithmetic, no? It's just that X number of natives are there, Y number of migrants are there. The X number of natives have a certain frequency. The Y number of migrants have a certain other frequency. You have to add them up. That's all, right? So, now Q naught into 1 minus N, these are the natives, correct? So, you could have a Q naught, you could be a Q. If you were Q naught and a native, or QM and a migrant, right? It's just a weighted summing, that's it, right? And so that is Q1. So Q delta Q is Q1 minus Q0, which is Q naught into 1 minus M plus M QM minus Q naught is equal to MQM plus minus MQ naught, right? This will cancel, no? Okay? M into QM minus Q naught. Okay, this is just a formula. But what does it mean? So if M is high, will the allele frequencies change quickly? Suppose Q naught is 1, okay, and QM is 0. Just take an example. This allele is, let's take it the other way. This is 1, this is 0. Allele is not there on the island. Allele is only on the mainland, and there are now migrants. Okay. So, what situation, under what situations would allele frequency change fast on the island? If M is very high, or? Right. If this difference is high, or if M is high, the change in allele frequency will be fast. That makes sense, no? Formulae, you can ignore them. You have to understand what they mean, okay? So now, is it possible to have an equilibrium? We talked about equilibrium. If there's equilibrium, what should be satisfied? So Qm basically will equal Q0. You can't solve it that way. But when they are equal, that is basically the equilibrium, right? And then there may be gene flow, but it will not cause any change in allele frequencies, right? So gene flow, suppose, let's just take an example. Let's not take such an extreme example. Suppose this is, okay, fine. Let's take 1 and 0, and M is 0.2. 20% of the individuals move, right? So delta Q in the first generation is 0.2 into 1. So in just one generation of movement, this frequency will go from 0 to 0.2, right? That's a huge change in allele frequency. That's a huge change in allele frequency. So migration is a strong force of evolutionary change. Very quickly, migration can alter allele frequencies, you know, very, very quickly. We'll talk about, maybe you're like, what is a strong force? We'll talk about that a little later. So now, this is one way to think about it, but that's the theoretical population genetics way. So I'm an empirical population geneticist. I work with data. So the thing is now, like, I don't know. Today something looks like an island or a mainland, but I don't know. Yeah. It does. But the net amount of difference that can be caused by M is very high. You'll see, for example, selection is also a very strong force. Very quickly, allele frequencies can be changed. If like all the small-beaked birds died, for example, the allele can be lost, right? So that's also a very strong force. But something like drift and mutation are could be relatively slow. It depends on the context again. Mutation is definitely very slow because mutation rate is very low, okay? So the ability of mutation to create new variations and the allele frequencies to change is really, really slow. It takes a very, very long time. In the sense that mutation rate is between 0 and 1, na? But for example, migration rate is between 0 and 1. Mutation rate is 10 to the negative 8. So it's very, very, very low, okay? So I was saying that, you know, this is what we think of as migration and how it changes allele frequencies in this very toy example. But how we might encounter this is a situation called population structure. So what is population structure? So this is a very clear example of an island and a mainland. But there may be other times where you don't know what the populations are. Are there populations? Are there many populations? Just one population? So how do you know? Like you go to some place, you see a bunch of lizards. You go to some other place, you see the same lizards. I mean, not the same, exact same lizards. See the same species of lizards. You go to a third place, same species of lizards. So what is the population then? Is India a population? Is Asia a population? Is South India a population? Is Western Ghats a population? What is a population? Right.
Any group of individuals are sharing a common resource that we can become that?
Well, not in population genetics. In population genetics, yeah, one way to detect population structure is to treat everything as one and see if it violates the assumptions of being one versus more. Okay, so basically imagine this is a space and you have for whatever reason there's a river here, okay, there's a mountain range here, okay, but there are individuals all over. So, if there was population, if there is one population, then this whole thing should behave and be in Hardy-Weinberg equilibrium, right? If there are three populations, you should have three subgroups, okay? So before we go there, I'll talk to you about something called the Wahlund effect. So what happens if you have these three subgroups? So actually, if it's three subgroups, we should have P1, P2, and P3, correct? Three populations, three different allele frequencies, right? But if you didn't know that there were three populations and you thought that these were all mixing up, what would be P? What would you think that P would be? Huh? No, but there's an exact. Yeah, it's the average, right? So you would think that P would be basically P1 plus P2 plus P3 by 3. No. 3 have equal number of?
Area or like
Doesn't matter. Doesn't matter. P is a frequency. If P was a number, you would divide by the frequency.
When you say P1 is the frequency of individuals and that patch.
Not individuals. Frequency of allele A
in that
patch.
So if that, say for instance, say that patch has some 100 alleles and the other one has another 100 alleles. For this
Frequency is usually less than 1.
Yeah.
Yeah.
Like out of, so you can eventually divide by 100. Not to talk in decimals. So in that case, suppose there's 100 and the second patch has 200.
Okay. That's the point. This is not 100, 200. This is normalized for the number. P is normalized for numbers. Does that make sense? No.
He's saying that the number of alleles are changing.
No, I'm talking about the frequency.
He's saying
100
and 200 and 200.
Huh.
It's just like the frequency of the allele will change, but the number of alleles will remain.
No, it could be different.
It's a biallelic state.
No. The genotype has, every individual has 2 alleles. That is true. But, okay, let's take an example before this gets confusing. Okay, 100 individuals here, 100 here, and 200 here. Okay, fine. P1 is 0.5. I'm telling you. P2 is 0.6 and P3 is 0.1. What does that mean? That means I went to the population. I found the A's and AA's and the AB's and the BB's. I got the observed AA, observed AB, observed BB. I use that to calculate expected AA. I use that to calculate expected P. You should be able to say this in your sleep now, right? How do you get at P? Take all the AAs, take all the ABs, divide ABs by 2, add them up, divide by the total number of individuals. That's how you would get P1. Do the same in population 2, do the same in population 3. But what I'm saying is, if you did not know that there were these 3 populations, it would be mixed, right? So the frequency of P would actually be P1 plus P2 plus, you would think it's P1 plus P2 plus P3 by 3. It's the average. Does this make sense? If it was a continuous population with all the alleles mixing. Okay? So what you would think then, actually the number of, this is capital P, okay? That is, who are these people? What are their genotypes? Huh? Small a. Okay, the small a people. What is the proportion of small a people? In these three, given that you know there are three populations, it should be okay. Pi, there are k populations, right? Pi squared. Similarly, capital H should be right? But what about the expected? This is the observed. Right? But what about the expected? Expected, you did not know that it is three populations, right? So P is basically just the allele frequencies averaged. P1 plus P2 plus P3 by 3, that's what this is, right? So if you try to, and so basically number of AA should be P bar squared, right? That's what you think. You think that they're all mixing, and so it would be P, the frequency, all squared. That's what you expect, right? So basically though, there's a difference between this and this. They're not the same. One is you take the average and square it. The other one is you take each one and square it, right? They're not the same. Okay? So if you want to look at what the difference is, suppose you do it in terms of Q bar minus Q bar squared, okay, is equal to 1 by K sigma Qi squared minus Q bar squared. I can just subtract 2 Q bar squared. That's the same thing. And then basically if you simplify this, again you can work it out, it ends up being what is that? Qi minus Q bar whole squared by K. Does that look familiar to you? It's a variance. Yeah.
Only if the size is comparable, or we are assuming?
No, it doesn't matter what this. The assumption is that the sizes are, the assumption for the model of FST, which I'll get to, is that the sizes are similar.
Expected value will either under the assumption that each of, like it's an infinitely large population and each
Yeah, of course.
And each patch is infinitely large.
That is, all assumptions in population genetics are based on the fact that they're infinite populations.
And each patch is also infinite.
Yes. I mean, every expected allele frequency you calculate is based on that assumption. Every single time you calculate expected allele frequency, it's based on that assumption. Okay, so basically what this is saying is that the difference between, what was this? This was assuming there's no population structure, a free mixing population, right? Which you thought, because you don't know. You can't, maybe there was a river which dried up, a mountain which was destroyed, right? You didn't see anything. It looks continuous. So this is what you thought. And this is the reality, right? And this number is positive. So basically for the homozygous types, AA or BP, the difference between not having population structure and having population structure is positive. For the heterozygote then, it's negative. So when you have population structure, you have heterozygote deficit, like inbreeding. You seem to have more homozygotes, but it's actually not the case. It's just because you have these distinct populations which have different allele frequencies, which you didn't take into account. Is this confusing? Everyone's looking a little lost. The only way to assess population structure is statistical. You cannot, you can't watch gene flow. It's not going to happen, right? Yeah. Sorry?
H bar will be 2 by k summation PIQI, or it will be 1 by k summation PIQI?
1 by k. So basically, if you have a high variance in allele frequencies, that is, if the P's in the different populations are very different from each other, you will expect to have less heterozygosity, right? If the allele frequencies are very similar to each other, you'll expect to have a little bit, not as bad heterozygote deficit, okay? So basically, what it boils down to is that Q bar is Q squared average plus VQ, the variance of Q, okay? Similarly, P bar is P squared average plus variance of P. And H bar, which is heterozygosity, is 2PQ, 2P bar Q bar minus 2 VPQ VQ. So the thing is, this is all very nice, and the problem is it's just too theoretical. So how do we actually quantify this? How do we, like I talked about lizards or whatever, how do we actually estimate whether there is population structure or not? It sounds simple, right? Suppose you had to actually do it. You want to see whether there's population structure or not. You just go to the field, you catch some lizards, you happen to know some locus which you can type, so you can get the allele frequencies. So what do you think? If you go to two locations and the frequency of the allele is quite different, you would assume that there is, population structure, whereas if the frequencies are very similar, you will assume that there is no population structure. So a way, right, population genetics is largely a product of three people's work, Wright, Haldane, and Fisher. And Wright came up with this concept of F statistics to look at population structure. Okay, so he talked about F statistics, where F XY is basically the correlation between gametes. You have three levels. There's an individual subpopulation and total population. Okay? So assuming the gametes were drawn from here versus the total. Okay? So basically FIT would be the correlation between, or basically it's like an inbreeding coefficient of what was the allele frequency here versus here, total. This is T. And what we use to quantify population structure is FST. So this is basically individual inbreeding. This is how inbred the subpopulation is versus the total, right? Is basically FST is 1 minus HS by HT. Why is it that? So basically, this is the heterozygosity, the relative heterozygosity in the subpopulation versus the total population, and 1 minus that, the opposite of heterozygosity is homozygosity or inbreeding, right? So basically, FST is 1 minus HS by HT, which is HT minus HS by HT. So to calculate FST, FST goes between 0 and 1. To calculate FST, you need to calculate heterozygosity of the total population, which you would calculate by the average allele frequency, right? Average P, average Q, 2 into average P into average Q, right? And HS will be the expected heterozygosity in each of those subpopulations, average over 1, 2, 3 or whatever, and again divide by the total. So we'll just, we don't have much time. We'll do a very quick example. I'll just write it out. We don't have to do the whole thing. But imagine if you have a population P1, P2, and P3, and again you have AA, AB, BB, and you have okay? This is just the numbers of individuals in those 3 subpopulations. Okay? So, to calculate anything to do with I, you have to calculate H observed. Well, you have 3 H observed. For P1, it is 4 by 10, right? What's the proportion of observed heterozygotes? In population 1, it's 4 by 10. In population 2, it's 2 by 10. In population 3, it's 8 by 10. Now, what is the expected heterozygosity in population 1? That is H expected P1. How to do that? Remember again, H expected in P1 should be 5 plus 2, right? By 10, 5 plus 2, 0.7. Yeah. So, okay. So, expected heterozygosity. Expected heterozygosity in P1 is 2 P1 expected, Q1 expected. Now, okay. So, P1 expected is 5 plus 2 by 10, 7. Q1 expected is 1 minus 0.7, 0.3. So, 2 into 0.7 into 0.3, right? 0.48. Similarly, you would calculate for P2, you would calculate for P3. And how do you calculate the total HT? HT would be P1 plus P2 plus P3 by 3 into Q1 plus Q2 plus Q3 by 3. I mean, right? Okay? That is if there were no boundaries. Yeah, you want me to repeat this last step? So, the second step you got, right? This is pretty important. So, observed heterozygosity would be based on, you calculate expected allele frequency in each of these subpopulations, and then you get the expected heterozygosity in 1, 2, and 3. So, HS would be the average of those three, right? HX1, HX2 plus HX3 by 3, right? Okay, now the last one, which is actually the easiest. Why is it easiest? Because you just ignore this. No. And calculate it based on these expected heterozygosity now. Right? So basically 5 plus 3 plus 1, 8 plus 8 plus 2, 3, 2, 3, 4, 5, 6, 7. 8 plus 7 by 30. Right? Got it? And then 2 into 8 plus 7 is 14. Huh? No, 15. 15 by 30 into 1 minus, 30 minus 15 by 30, whatever. Yeah? So that is the HT, expected heterozygosity in the total population. Got it? Anubhav, want to say something? Yeah.
So HT is the heterozygosity or expected heterozygosity of the total population.
Yeah.
Right?
Yeah.
Given this is
Given that you don't know that there are populations.
And HS is the
Of
considering the subpopulation
expected in each of the subpopulations.
and then an average,
Yeah.
so like the total heterozygosity of the total, but considering that there are
Yeah.
participants.
Yeah. Okay. And HI, why is HI? HI is observed because if there's inbreeding, HI is basically reflecting inbreeding. If there's inbreeding, the number of heterozygotes will go down. But inbreeding will not impact the expected level of the estimate of allele frequency, right? So when you're talking about, you don't want your local inbreeding to impact the global FST. So just to very quickly talk about FST, FST ranges from the order of 0 to 1. FST is also proportional to divergence time. If two populations split a very long time ago, then FST will be high. In fact, we won't have too much time to talk about the coalescent, but if you model this in the coalescent, when you have 2Ne to 4Ne generations, Ne is the effective population size or number of individuals, FST equals 1. So that's how long it takes for the two populations to become completely different. FST is equal to 1 means they're completely different. Okay? Yeah, it's very small. So it takes a lot to get FST high. Okay? And by the way, yeah.
When I'm saying it is 0.0, so I'm ruling out that these are three different.
Yeah, I would say, yeah, exactly. So if I was an empiricist, which is what I am, I would say FST is very low. So FST, for example, between human populations, anyone knows, it's on the order of about 10 to 15%. Any two human populations, FST is about 10 to 15%. Okay, but say FST between two tiger populations is on the order of 20% or 30%. Within India, like Western Ghats compared to Central India, FST is 20% or so. Okay, so much higher in some sense compared to humans if you say compare between Africa and Australia. Okay, so just to tell you also that FST is equal to 1 by 1 plus 4 NEM. okay, where NE is effective size or the number of individuals and M is migration rate. So imagine if you have two very, very large populations, FST will be small. It's difficult to get very high FST if your population size is very, very large. Similarly, if M is small, FST will be low, inversely proportional, right? If NE is very small, if you have a small, two small populations, you can jack up FST very quickly, okay? It's inversely proportional. So both of these factors affect FST. So by the way, this is one of our problems when you say, when people say, oh, just estimate migration rate. We can't, we cannot, we can only estimate the product of NE and M because they can't be disentangled from each other. You have to make some assumption about NE, then you can say what M is, but otherwise it is very difficult to estimate M, as in the migration rate, okay? You can estimate the total effect of migration in terms of numbers by this composite thing, NEM. Oh my gosh, I don't have much time left for drift. But they're all kind of the same thing, you know. Inbreeding, population structure, and drift. So drift, I think it's easier to think about drift from a qualitative perspective first, heuristic perspective. Drift is sampling, okay. So remember you had that bag where you were sampling the beads, right? So imagine if you had 3 colors of beads, red, blue, and white. Okay, so let's have only 2, red and blue. And the frequency of red is 0.8, the frequency of blue is 0.2. What is the probability of pulling out 2 reds? Huh? Pull out a red and pull out a red. 0.64. So when you pull out 2 things, you could pull out 2 reds, 2 blues, 1 blue, 1 red, right? And this is easy, it's just a binomial process, right? But the same thing is true if you pulled out 50 alleles, right? If you pull out 50 alleles, will you always get exactly 25 reds and 25 blues? No. You will get a distribution, right? And what is the probability, if you have 50 trials, what is the probability of having exactly 25 reds and exactly 25 blues? You guys know this. It is P plus Q raised to N, right? And I'm saying, I want you to tell me the probability of getting N by 2 reds. This is red and this is blue, and N by 2 blues. Yeah, exactly. What? NC? Yeah. NC N by 2, P to the N by 2, Q to the N by 2. Right? Remember this? From way back in 9th standard or 10th standard. Right? So, if you do 50 trials, some of the times you will get this. Other times you will get 49, 51. Other times you will get 2 and 98 also. So the thing is, drift is again a theoretical concept where we're thinking that imagine if we sample that gametic pool not once but thousands of times. And from each of those times, we estimate allele frequency. Suppose I pulled out 98 reds and 2 blues. What is my allele frequency estimate? It will be very different from when I pull out 50-50, right? It's the same bag. It's the same bag, but my probability of pulling this out, it has a certain probability. This is very low. But by chance, I could land up in that bin. By chance. So that is drift. The fact that every time you pull out alleles, you pull out a finite number, not an infinite number. If it's an infinite number, it's okay. You pull out a finite number of alleles, there will be a bias. Sometimes you'll pull out more of one than the other. Okay? And basically, and now I'm feeling very hot, drift has the strongest effect when the population size is very small. Stochastic effects like drift are highest when the population size is small. So what is the effect of drift? What will happen with drift? Alleles can be lost. Actually, because of drift, you lose heterozygosity. Okay? And drift is another form of inbreeding. So we can think of drift. Imagine that there was an inbreeding coefficient of T in the previous generation. The inbreeding in this generation okay. Suppose your population size is n. You have 2n gametes. The chance of pulling a specific gamete is 1 by 2n, a specific gamete. The chance of pulling that same gamete again is 1 by 2n, exactly same gamete again. But there are 2n gametes. So you could have pulled out the first gamete and it's inbred, the second gamete and it's inbred, the third gamete and it's inbred, the fourth gamete and it's inbred. So that's a OR. So add this 2n times. So that's into 2n. So 1 by 2n is the chance that in this generation, you will pull out 2 gametes which are identical by descent. Okay? And so either they are identical by descent in this generation, or they were already identical by descent in the last generation. Or, so what is the probability that they were identical by descent in the last generation? That was Ft, right? But they were not identical by descent in this generation. The probability they were identical by descent in this generation is 1 by 2n. So those which are not identical by descent in this generation is 1 minus 1 by 2n, right? But they were identical by descent in the last generation. So that's into Ft. Should I explain that again? Okay. So identity by descent. That is, I pull out 2 alleles which are exactly the same. So given that a population is of size n, there are 2n gametes, right? Each gives 2 gametes. The whole point of drift is it's a finite population size model, okay? So you got to get in, we have to get our hands dirty with n. So far we were talking about infinite population sizes and we were happy. So now there are 2n gametes and I'm going to, it's not just that, you know, the same individual has 2 gametes. If I pull both of the same individual's gametes, that's not identity by descent. The same gamete has to be pulled again, right? So the probability of pulling the same gamete once and again is 1 by 2n into 1 by 2n. It's sampling with replacement, remember. You pulled the gamete but you threw it back in somehow again, okay? But there are 2n gametes. So the first gamete I pulled could be identical by descent, or the second one could be identical by descent, or the third one could be identical by descent, right? So this can happen 2n times, right? So I multiply by 2n. So the total number of gametes, the probability that gametes are identical by descent is 1 over 2n. Okay, yeah.
You are pulling out only 2 gametes.
Yeah.
But you said we are pulling out 2n.
No, it's either you're pulling, so it's either this, so there's like 2n gametes. It's either the first gamete which is identical, or the second gamete which is identical, or each gamete has a chance of being identical to itself. You could pull it out and it's identical to itself, right? So you're doing this 2n times. I mean, each gamete has a chance it can be identical, can be pulled out, and and it's identical, same thing could be pulled out again, right? Yeah. Because I'll get to it.
Because that was what you were referring to in inbreeding.
Yeah, but they're all heterozygosity and inbreeding are flips of the same thing, as is drift in some sense, because allele frequencies are going to change because of finite population size. Okay, the heuristic explanation of the stochastic sampling is really how drift plays out, but how do we calculate? That's an expectation, isn't it? I can give an expectation of allele frequencies if I had 50 alleles, I mean 50 population size. This, oh, I lost it, but this is equivalent to population size, isn't it? However, if I wanted to actually quantify change in allele, this is an expectation, it's a distribution, but I want to quantify change in allele frequency, how do I do it? The change in allele frequency will occur because of inbreeding, right? Because you have a small population size, there's a probability of inbreeding which will change allele frequencies, okay? So now, we have 3 minutes left, no, 1 minute left. So yeah, so we were talking about f of t plus 1, right? Now f of t plus 1 is inbreeding in this gen plus inbreeding in the past. Got it? Yeah? Inbreeding, oh, there was already inbreeding, or inbreeding happened in this generation. Okay. Inbreeding in this gen, we happily decided and agreed it's 1 by 2n, right? Or it happened in the past. The probability of inbreeding at generation t is ft. That is the probability that it was inbred, an allele was inbred in the past. But it did not get inbred in this generation. Okay? So this is ft plus 1 is equal to 1 by 2n plus 1 minus 1 by 2n into ft. That's a recursive equation. And if you write it out, basically, by the way, also 1 minus f is what? ht. Okay? Heterozygosity. Not being inbred, being outbred, right? So basically, you can work this out as well. If you do 1 minus ft plus 1, which is ht plus 1, is equal to 1 minus 1 by 2n into ht. So ht is 1 minus 1 by 2n raised to T H naught. It's just the same thing. So heterozygosity will always be lost under drift, right? Because HT will always be less than H0. The higher the N, the lower the rate at which heterozygosity will be lost. The lower the N, the higher the rate heterozygosity will be lost. Okay, we'll stop. We can quickly revise this the next class, which is on Friday. But Friday will be a bit easier because it's selection and mutation, which are both easy. But yeah. I think Thank you.